Good morning, everyone. Welcome to another REGARTIS webinar on endocrine disruption. This time, my colleague Adam Jonas will focus on endocrine disruption basics. I sincerely hope this webinar will be useful, and I'm looking forward to also your participation. For this participation, I would like to set a few, let's say, rules or maybe guidances. Chat should be open to anyone, so are questions and answers. I'm not sure if we'll be able to manage any extensive chat communication, but we will definitely manage questions and answers. After the initial presentation, we will go through a discussion. In this discussion, we will give priority to people who would like to speak, as this is supposed to be also a discussion platform. You aren't able to unmute yourself by default, but if you raise your hands, I can give you the permission to unmute yourself. This is only for the cases when multiple people would be talking over each other. This is to prevent some this sort of like traffic commotion. And also, you can pose the questions in questions and answers. So the priority would go through the microphones, then questions and answers, and then chat as well. So this is me giving the floor to Adam. Welcome, Adam.
Thank you for the introduction. So let's go for the presentation. I'll share my screen for you with the presentation. So this presentation is about endocrine disruptors. This time it's about basics. And my name is Adam Jonas. I'm ecotoxicologist and toxicologist also. I also did quite a lot of regulatory work in biocides, plant protection, around different regulation in Europe, also some of US, New Zealand, Australia. And today I prepared for you some news, what happened from the last presentation. So we are already in the content of the today's presentation. Then I will talk about regulatory overview to kind of do some bridge into the endocrine system informations. Within the endocrine system, I'll talk about its modalities, so basic modalities, pathways. I will explain you about the hazard and the risk in toxicology, endocrine activity, then some tests, assessments, and at the end I will show you how now I do the classification assessment for endocrine disruptors. So the news of the day is actually that yesterday was published final guidance on endocrine disruptors. It's actually full CLP guidance, so there is everything how to deal with the CLP. So there is also all the previous hazards. There is endocrine disruptors and also this fate hazards. And there will be this ECHA presentation on 21st this month. So I would be interested what they will be saying there. Another news, there was a 6th annual forum on endocrine disruptors. So this is a forum held by European Commission. It was at the end of October. So I prepared some basic informations from this forum, so you don't need to look into it. But you can, of course, it's interesting and it's on the web. So what was discussed there were some plans, achievements of ECHA Commission. There was a presentation about beautiful research done on endocrine disruptors by great European scientists and also some abroad scientists. And first presentation, there was about one substance, one assessment. So this is a concept which is more and more heard about. So it's basically that when we have in Europe one substance, that it will be assessed on once in common. Also, it was said that there will be common data platform for chemicals, which will be shared by Commission, ECHA, EFSA, EMEA. For me, this was first time which I heard about European Medical Agency to be actually joined in this one substance, one assessment. For me, it was surprising because for pharmaceuticals, if the substance is endocrine active substance, then you are required to do risk assessment for this substance, which is in contrast with the ECHA EFSA, which they required hazard assessment. So I'm interested how this one substance, one assessment will work in this case. I will talk about the difference about hazard and risk assessment later on. So this is just ahead. So also environmental agency and EU OSHA will be joined in this. Then there was a presentation about evaluation so far in ECHA EFSA. So I recommend this presentation to have a look on. And also there was information that there will be joint report on plant protection and biocidal products on the ED evaluation in summer 25 published. Sorry. So about the guidance, it was also discussed in this forum. So the guidance was early published in November, and it really contained also the PMT PBT guidance. The guidance is published with some open topics. So basically, RAC will be doing the assessments, and based on the lessons learned from this assessment, the guidance will be updated in future. And this is the link here where you can download the videos and also the presentations from this forum. Okay, so we now go into the regulatory. So this is the delegated regulation which brought these hazard classes into the CLP. CLP classification, labeling, packing regulation. It's a central piece of regulation which influence all the other chemical regulations. So it's a very important one. It's almost 2 years in force now. And this new hazards, it's not only endocrine disruptors, which I will talk about today, but it's about also this PBT, PMT, especially the mobile, is a very new concept. So last time, when we were having the webinar, you could hear from a lawyer specialized in these regulations, chemical regulations, that when you have a batch of a substance newly produced, so from 1st May 25, you need to have already correct labeling according to these ED classes. So is there enough time now? No, there is not enough time. Basically, you have few months to do the classification and to get prepared. What's actually the impact of the classification? So in current REACH, the ED classification leads to the inclusion on the SVHC list, so substances of very high concern, and this leads later to the authorization and possible restriction. In Water Framework Directive, endocrine disruptors goes to the priority list. And in plant protection biocides or coformulants, impurities can actually trigger ED classification. And also you should be careful of what you have, because it's difficult to change impurity when there will be a threat of authorization of or restriction of this rich substance. In cosmetics, it's unlikely to maintain ED substance in shampoo or fragrance or whatever. So what ED hazard classes are now in CLP? So it's endocrine disruptors for human health, environment in two categories, known or suspected endocrine disruptor. And there are three criteria for ED. There needs to be activity, so some change in hormones, for instance, adversity, so some negative effect, and there needs to be a link between those, so the activity is actually leading to this adversity. And the classification is made based on the total weight of evidence using expert judgment. So now we go into the endocrine system. On the left, on the picture, you can see all the tissues, glands, which are involved in the endocrine system, the main tissues, not the hundreds other hormones in the guidance. But you can see hypothalamus, brain, pineal gland, pituitary gland. So this is communicating with other glands within the body, which then communicates with the other tissues. When you would like to read here on the right the definition from the guidance. So the endocrine system consists of hormone-producing tissues and their associated hormones that regulate the functioning of the organism. Maybe even nicer definition is below this picture. So endocrine system. So endocrine system regulates various bodily functions. It regulates it through the secretion of hormones. And these hormones are kind of chemical messengers that travel through blood to target organs. And these hormones control processes like growth, metabolism, reproduction, stress, or overall homeostasis. Now I go back to the definition in the guidance. So they define endocrine disruptor. Endocrine disruptor may alter one or more functions of endocrine system, e.g. hormonal synthesis, transport, signaling, regulation, or metabolism. So that's basically what all our hormones are doing. And I put here also part of the medical article. So they say that hormone replacement therapy is used when insufficient hormone production by endocrine glands can be replaced by identical hormones processed synthetically or biotechnologically. So there is some concentration in body, and this concentration is considered good. But if you have low concentration, then you can use these artificial hormones to balance the system, or actually to also lower the concentrations of the hormones if they are produced too much. So it's not only chemicals which can unbalance, but also like malnutrition or some disease which can unbalance endocrine system. So its modalities, so because of the limited knowledge and ability to test endocrine pathways, these modalities were chosen to be investigated in the old ECHA guidance in 2018. Also in the new guidance, these are the main pathways, main modalities which are investigated. So this is kind of similar. You can see here a lot of endocrine active substances. So do you recognize which are the bad guys, which are the good ones? So don't worry if you don't recognize the substances. Also don't know if they are good or wrong. It's our human nature to judge what's good or bad. So for instance, this is a carcinogen substance. It's responsible for, or at least in the old literature is said that it's responsible for 1/3 of the breast cancers around the world. But in this new Nature article, they just published in 2024, they said it's actually prominent in the development of breast cancer and also other cancers. And what is the substance? It's estradiol. Estradiol, so that's our natural and endocrine substance and a natural hormone. It's a very important for body. It's important for development and reproduction. The humans cannot exist without estradiol, basically. So it's not easy. Even our own substances which we have in the body are carcinogenic, dangerous. It's always about the concentration, and there is definitely a cost for reproduction, which is this carcinogenicity. It's quite severe. On the right, you have ethinyl estradiol. You can see that the substances are very similar in structure. I will talk about it on the next slide. Then you have some other hormones, and actually the substance with the better reputation is here. It's a bisphenol A. Okay, so this is how the hormones works. So they works as a key to a receptor. So actually the structural similarity to hormone cause the endocrine activity of these substances, which you could see, you could saw on the slide before. So the for hormones to influence the target tissues, they go through the bloodstream and then binds to the receptor within the target tissue, and then some actions are happening. So the hormones do the influence like this. Thyroid modality, that's actually from the scientific point of view, my favorite modality somehow, because it causes difficulties and it's a quite complex modality, I would say. So thyroid modality, it contains feedbacks, balancing the concentration of hormones within body. You could see here hypothalamus, so brain release thyroid releasing hormone, which goes to the pituitary gland, which produce thyroid synthesizing hormone, which goes to the thyroid tissue. And if there is a lot of TSH, then more T4, T3 thyroid hormones are produced. And if hypothalamus think that it's too much T4, T3, again, it releases more TRH, and that translates to less TSH, and there is less thyroid hormones. This is not used only for balancing some external thyroid chemicals. It's for instance when there is not enough iodine in food, then the thyroid gland will be kind of growing to be able to catch more iodine, and TSH will be more synthesized to make T4, T3 in higher concentration in the body. So for instance, I can tell you one example. So in one region, they banned iodine disinfection for milk tanks. So this disinfection was used to disinfect these containers of milk. And as a result of this ban, most probably because iodine is evil substance which cause toxicity and also endocrine disruptor, it has an impact on the population in the region because they start to be deficient in iodine. So these trace amounts of iodine in the dairy products, in milk, they actually give into the bodies the necessary iodine. So it's interesting example how the ban of basically biocide in this case can influence the well-being of people. The thyroid is actually influencing everything. So thyroid system impacts metabolism, weight, development, heart, intelligence, behavior, immune system, spermatogenesis, male, female reproductive organs development, number of offsprings. So also the disruption in this system, it leads to very severe conditions by disease, by disruptor, by anything. Thyroid modality, it was actually responsible for most of the banned substances in plant protection and biocides. This is because the estrogenic, androgenic substances are more easily catched because they also can be carcinogenic, for instance, and also by reproductive effects, developmental effects. In the old battery, it was much more easier to see this, and this was already enough information to restrict these substances before the endocrine disruptors came into the place. For thyroid, it's maybe more difficult to find out about the activity and effects. So as I was telling you before about the lock and key, like this is only this mode of action, thyroid receptor agonist and the receptor antagonist. But the mode of action which can disrupt the thyroid it's much more TPO, NIS inhibitors, deionization inhibitors, TH clearance, and so on. So it's a lot of mode of action within this thyroid modality. I go to the estrogen pathway. So there was research done on ethinyl estradiol in water, and it was shown that this ethinyl estradiol pharmaceutical induce feminization in the fish and collapse of the fish population. This was beautiful research done by KIT in 2017. So what they did is that they went to the beautiful Canadian nature, to the huge lake, and started dosing this lake with the ethinyl estradiol 5 to 6 nanograms per liter. And what they saw is that the fish were turning female, and was a decreasing amount of males within the system, and at the end the fish population collapsed. Interesting research. I would say that there was follow-up research which shown that actually the populations get well with the time again, because there are some streams, some rivers going into the lake, so the fish populations, there was recolonization and so on. So it was not stable effect. And also 5 to 6 nanograms of ethinyl estradiol. In past, I did some research on European wastewater treatment plants effluents, and I've never seen so high concentration of estrogen substances within direct effluent from the wastewater treatment plant, as was used in this article. So fish feminization was discussed for decades in ecotoxicology, and I think the pharmaceuticals like ethinyl estradiol were main reason for potential adverse population effects in nature. Then we have non-EATS modalities, and this is because the CLP guidance actually is opening the scope to all other modalities. So we can read about that there is about 100 hormones produced by other tissues than standard 51s and so on. So I would say that we have good understanding to estrogen androgen pathway, and we have good testing methods. We have somehow limited knowledge about thyroid pathway and limited testing abilities. But for these other pathways, we have limited knowledge, we have limited testing strategies, and we don't have the OECD test to test these pathways. So it's tricky, it's tricky. But I understand when substance is clear, I don't know, corticosteroid, then it's probably endocrine active substance. So, but it will be tricky. So with these two lions, I would like to tell you about how the substances are regulated in regulatory toxicology. So you can regulate either by hazard, that's for substances like carcinogens. So the thinking behind is that actually one substance can binds to DNA or disrupt DNA somehow, and this one molecule of this substance can do the damage. So then for these substances, you don't have the threshold, and even one molecule can be dangerous. So then you regulate based on hazard. You are not looking into the concentration of the substance. You just see the dangerous property of the substance, and you are trying to ban this substance. On the other hand, there is also risk approach. This is, for instance, for acute toxicity. So we know in the low concentrations, there is no effect of the substance. And when the concentration is high, then there is a mortality occurring. So very severe effect mortality. But because we can manage the risk, we can manage the concentration which we are using to be using only safe concentration, the risk is controlled and the hazard of the substance is not happening. In CLP for endocrine disruptors, we are using the hazard. So basically, it's saying that we cannot control by concentration the risk. On the other hand, in the environmental risk assessment of pharmaceuticals, you have the risk assessment for hormones, directly the hormones, the strong substances. And also in other parts of the world, you have the risk assessment for these substances for endocrine disruptors. Why is it like this? It's because endocrine activity is actually not a unique property of a chemical. It's actually quite common. So some essential nutrients like iodine or vitamin A have this property. If you don't have enough iodine or vitamin A, there are serious issues like that, for instance. But if there is too much vitamin A, there are malformation in development. So again, serious issues in the high concentration. Food and beverages, hops, soya, they have estrogenic activities. So in soya dish, you can have 1 gram of phytoestrogens per 100 grams of the soya dish. So very high concentration. So I would recommend not to give to small boy only soya dishes. And it's not so unique. There is at least 300 plants with phytoestrogens. Also, for instance, clover, so common plant on meadows. It was shown that when you have sheep which are eating too much clover, they have reproductive issues. There was also case study with female workers in the factory where they were processing a clover, and again there was some reproductive issues seen in these workers. Then mustard, there is bisphenol F in it. Then endogenous hormones, we already talked about it, estradiol, testosterone, pharmaceuticals, ethinyl estradiol, and man-made chemicals like bisphenol A, bisphenol F. Here you can see endocrine activity. So that's from one of my favorite articles. It's actually a fish model. You can see on the y-axis the fold induction, so the effect, and here you can see the concentrations. These are the pharmaceuticals, ethinyl estradiol. So you can see very low concentration to cause the effect, and also the effect magnitude is very high. Then you have natural hormones. So it's a few orders of magnitude lower effect. On the other hand, this is fish. So in human cells, for instance, the ethinyl estradiol has the same effects as natural hormones. Fish are very sensitive to ethinyl estradiol and synthetic estradiols. Then you have food. And you can see that in food, the concentration which cause effect are like 6 orders of magnitude different. So on the other hand, when you eat food, you can eat this 1 gram per day. So it's quite a high exposure. Substances in plastic, very similar concentration ranges as food. But usually we don't eat so much plastics as food. So there is some difference. And last are some banned plant protection products. You can see again the similar concentration ranges. You can do some further reading on the topic. There is an article from wise people from the universities, which is saying that human exposure to synthetic endocrine disrupting chemicals is generally negligible compared to the natural compounds with higher or comparable endocrine activity. And another article here is from Switzerland, from their food safety authority, and they found out that there is actually natural occurrence of bisphenol F in mustard. They also did a risk assessment for this bisphenol F. And what they conclude, you can read this risk assessment on EFSA pages. What they conclude is that it's safe to eat 80 grams of mustard with 8.35 milligrams per kilogram bisphenol in it per day. So they did a risk assessment on a substance which we cannot do a risk assessment. In CLP, it's actually regulated by hazard. So then if you see the effects, this compound bisphenol F is directly for restriction, of course. So I did some recalculations, and I was calculating the amount of bisphenol in this mustard to amount of bisphenol in plastic bottles. I used bisphenol A because I didn't find the concentration in paints where the bisphenol F is used, but bisphenol A and bisphenol F are in the same range acting, same concentrations, and the effects are also the same. So to compare, I calculated that you would need to eat around, on average, 100 plastic bottles a day to have the same intake of the bisphenol as from the mustard. So you can see the difference here between the risk assessment approach in case of mustard and the hazard assessment in case of plastic man-made chemicals, where on the hazard assessment this substance is banned, restricted, and in risk assessment you find safe concentration which you can use. It also illustrates the feeling that the natural substances are safe, whereas the man-made chemicals are evil. So there are the two things here happening. I think the concentrations are, the calculations are about correct, because also in this last article on the page, you can see that the exposure to bisphenol A is around 3,000 times lower than exposure to phytoestrogens in normal life. So another article, here it is from the German authority, BFR, and they are saying that actually there is evidence that they are threshold for the adversity for endocrine compounds, and that there can be sufficiently protective reference values derived, which would protect public from these effects, endocrine effects. Again, this is something which is not happening in CLP. The CLP goes in totally different way. In CLP, we will be restricting the substances which have effects in very high concentrations. So this is a battery of tests which is used in biocides plant protection to test endocrine disruption. In REACH, we don't have any requirements yet, but we can guess that for high tonnage substances, this will be probably the requirements there. So level 1, that's literature search and assessment of available information, QSARs. So that's basically what you are required to do by this CLP guidance now. Level 2 is in vitro studies. Level 3, mechanistic in vivo studies. Uterotropic assay for estrogenicity, Hirschberger assay for androgenicity. Then level 4 study, so it's more complex, more expensive, more difficult studies. So developmental neurotoxicity study, that's important for thyroid pathway. And then level 5, the most difficult, complex, multi-generation studies, as example, extended one generation reproductive toxicity study. The interesting thing is, for instance, that if you fail on this in vitro assay, then just up to recently, there was nothing else to do than jump directly to extended one generation reproductive assay, which is really, really expensive study. So, number of evaluated substances of ECHA and EFSA combined in between 2018 and 24. Basically, they had 51 inconclusive substances. So I think that they can be candidates for category 2 under CLP. So because you don't need to be completely conclusive in CLP, you just can say it's category 2. And from the evaluated ones, it's basically around 1/4 endocrine disruptors, mainly the thyroid pathway. And for ECHA, ECHA evaluated around 30 substances, from which 21 were inconclusive. So we can think about like how they are going to manage the thousands of rich chemicals. It's not so fast. And also, if there will be the REACH update, I think it will be difficult to find space in the labs to do the experimentation. In the guidance, there is some new concepts. One of them is AOPs, so adverse outcome pathways. So I wanted to explain you this concept. It's used instead of the mode of action. AOPs basically kind of are not using the concentrations to describe more or less the mode of effect, mode of action. I will show you an example. So you have always the molecular initiating event. So that's the how, for instance, how the compound binds to the receptor. Then you have several key events. It can be much more key events, which at the end leads to the adverse outcome. This is used in the guidance to make link between the activity and adversity and the initial molecular initiating event. So it's to bind this activity, adversity, to do this plausible link. And also the industry or anyone who is doing the assessment is required to do the mode of action in this way in the AOP's nomenclature to link this activity and effect. So as I said before, the data requirements in CLP doesn't have data requirements, but you need to assess the current data. So I have a recent example where there was 200 hours spent on a medium size data set for each chemical. In biocides, pesticides, easily 300 pages documents are done with this extensive Excel file. Systematic review can take 1 year of teamwork, so it's can take a lot of time. So how to plan the classification assessment to comply to be in line with the regulations? You need to do the assessment of endocrine activity or adversity for your substances to be in line with the regulation. And I think also to prevent some surprises like loss of coformulants or fragrance, you should check also what we are putting into your products, because when suddenly some substance will be restricted, loss, you should have some replacement. And I think also you don't want endocrine disruptors to be in your shampoos and so on. So my recommendation would be to try first light assessment, looking for ED indications with literature search, and later, if needed, you can do the full assessment with literature review. And we are going almost to the end. This is how I do the light assessment myself now. So I first screen the available ED lists. So if ECHA EFSA, for instance, didn't already consider the substance which I am screening endocrine disruptor, so then if the substance is on the list, then the screening stops, basically. Then I screen the chemical databases for endocrine hits. So if I see something about endocrine in these databases, then I make some QSARs. So you can use, for instance, OECD Toolbox, Vega system, Oasis, or Tox21 US system for calculations of QSARs. So the QSARs, for instance, they compare the structure to the hormones and tell you if the structure is very, very similar, that there is a high probability that the substance will be also endocrine active. Then I review literature, and here it can get really time consuming because you can have substance which has like 50 articles published about it, which you can screen quite fast. But I also had substance in the past for in biocides, pesticides, which had actually 20,000 articles published about. So then it gets kind of time consuming, you know. And I think here you need to search for CAS and chemical names, because if you target like a hormone endocrine disruptor, you can miss these non-EATs because, for instance, when I had some article about retinic pathway, so vitamin A, basically, I didn't consider vitamin A as a hormone. So if you search hormone, you would not find these articles. The most important for the assessment, I think, is to read the available studies on the substance and check for the ED indications. So for each modalities, it's best developmental, reprotoxicity, chronic studies, full life cycle assessments, basically. So on conclusion, so remember that CLP applies hazard assessment for endocrine active substances. And for me, it would be nice if you would remember that for well-functioning endocrine system, you need a balanced concentration of endocrine active substances, and that the natural active substances have actually similar properties as unnatural ones. So they don't know if they are natural or unnatural. Our body also don't recognize this. And the disbalance in hormones, too much or too little concentrations, leads to very severe effects. So it's not easy. So there can be some adverse effect, but there are also it's chance that there is no adverse effect at the end. And this is the end of the presentations. And now we will look into your questions. Thank you for your attention. Question from Yelena. Exposure to low levels of toxic metal lead is known to affect human health, and its potential role as an endocrine disruptor has been reported. Okay. Okay, so the lead it's a possible endocrine disruptor. Lead is already quite toxic material. It says in the question that it's not on the list. So I think now it's on self-classification of the people who are dealing with lead. So you should look into all the data, and you should evaluate if the lead is endocrine disruptor or not. So you really need to screen the data and decide by yourself, or to ask someone to help you, like myself, and say if the classification is needed or not. So at this moment, the ECHA let it on the applicants to do the classification. If the let is in some CORAP community rolling plan, then this will be assessed by RACT and RAC at ECHA, and the harmonized classification will be decided by RAC. But I think it's good for all applicants to prepare ahead to do their own classification and assessments to know if you have actually endocrine disruptor or not. So
thank you, Adam.
Thank you.
We can return to it if there's any more input needed. But there's another question in the Q&A at the moment from Juan. If you have a coformulant in a biocidal product, for instance, tensile surfactant, what would be done to assessment for conclusion that the ED criteria is not met? Is full literature research enough?
So if you have a coformulant in surfactant, well, there's also the concentration threshold, which is in the guidance. On the other hand, the concentration threshold can be basically lowered or increased based on how much the substance is considered endocrine active or endocrine hazard. So you cannot probably only say that it's below this threshold concentration, and it's not making the whole mixture endocrine disruptor. I think you need to do, for assess this substance, you need to do at least this light assessment. So you need to check the literature, you need to do the QSARs, you need to check the available data behind to be sure, or reasonably sure, based on the available information, that it's not endocrine disruptor. So as I was talking about this light assessment, I think you need to do at least the light assessment to decide if the substance could be endocrine disruptor or not.
Meanwhile, Yelena returned with a follow-up. If she understands well that the producer or supplier should assess possible ED properties and propose new classification to ECHA.
Yeah, so you can self-classify, and you can also directly ask ECHA for a harmonized classification. That's of course possible. I think it can be quite tricky for somebody using the substance if this substance is proposed for harmonized classification as endocrine disruptor. So it's also a reason why you should probably check your coformulants and additive by yourself, so you are not surprised when somebody actually asks for harmonized classification as endocrine disruptor at some moment. Yeah, so anyone can, so producer supplier should assess possible ED properties. If you believe the supplier producer, then you can believe, but if you want to check yourself to be sure that this substance will not become endocrine disruptor, suddenly you can also check yourself for this.
Okay, thank you, Adam. There is another question from Anu in chat. How to avoid using the endocrine disruption substances or endocrine disruptors, I guess, in our formulation? What regulatory actions need to take place for the company if a supplier or customer asks about the presence of endocrine disruption in the product?
Again, you need to evaluate to do the light screening to find out if the substances in the formulation could be considered ED. There is not another way than to go to the data and to assess. Like currently, the clear endocrine disruptors are on the list of ECHA, but now CLP asks you to evaluate by yourself the substances. So you need to evaluate to be able to say if it's endocrine disruptor or not. So I see also here a question about testing requirements. So do you have a view on the animal testing requirements for ED classification, or your view is sufficient to stop with level 1, 2? So that's QSAR in vitro assays, if there is no endocrine activity. So from my point of view, the QSARs are informative. For me, if I do the light screening, the QSARs and eVitro, it informs me that there could be some issue, and I look for the issues indicated by QSARs in the data, the animal data, because I would say I hope in the future there will be new approach methodologies which will be very complex, and we could avoid the animal testing in future. But up till now, for instance, for the thyroid effects, I don't think that you can really have 100% 100% belief to the QSARs that there is no effect on thyroid pathway or on other pathways. Also, sometimes I saw cases where you have positive in vitro effects, but in vivo later on there were no effects. The whole body somehow could handle the activity which was seen in vitro. They are beautiful also for neurodevelopment. They are in pipeline, nice QSARs, and I hope this will be possible to be used. But at the moment, I think the data set of the substance, like developmental studies, reproductive studies, are really the important ones. But if these studies are not available, CLP don't ask you to do any study. So you are not required at the moment to do any study, animal study. When there will be the future update of the REACH, there will be the requirements for REACH substances to do this testing. But up till then, you don't need to test anything. Okay. So I see here some pharmaceutical questions. So what do you think of topical use of estradiol with progesterone for a long time in treatment of menopause? It's how to approach. So the treatment of menopause, also the food containing phytoestrogens is recommended for this treatment. And on the other hand, the substances are estrogenic, estrogen active, they would be considered in CLP as endocrine disruptors. Possibly they are carcinogenic, but the treatment is good to do. So there are kind of trade-off in this. For me also, it would be quite funny if you have a medical pharmaceutical like ethinyl estradiol, and then you would have a rich chemical which is considered endocrine disruptor, and this endocrine disruptor will be banned, and you would need to stop using this addition into your pharmaceutical ethinyl estradiol. That would be quite funny, because estradiol is much more, it's like 6 orders of magnitude more potent than any of these rich chemicals. And also in the formulation, you would have probably only like 1% of the addition. So this is kind of not balanced, I think, at the moment. And I think we need to use the medications. So I don't know what to say. Like we need as a humankind, we need the corticosteroids, we need the ethinyl estradiol, progesterone, and so on. I don't know if this again answered the questions, but that's my answer. So I see question from Elena. So under REACH at the moment that there is no requirement. What happens if you screen some effects that might be ED alerts? Do you classify as ED? So if in the data sets, if there is really a clear picture that the substance is for sure endocrine disruptor, then the applicant should classify, yes. It's a question if it can be really so strong, the data set at the moment, but if you decide that the substance is endocrine disruptor, you should classify.
Elena also follows this question up with another part. I will read it out loud. They think that no studies are specific for ED under REACH at the moment, but there is still the potential to trigger the potential trigger of OECD 443 at lower tonnage band. What do you think about it?
Well, some substances with which are produced like 1,000 tons a year, they have quite big data set. So they have the reproductive studies, they have developmental studies, they have extended one generation studies. So from these studies, you can already see the disruption, and there are also measured thyroid hormones very often in these studies. So weight of thyroid. So with high tonnage substances, you can do quite decisive assessment. You can really decide. But in lower tonnage bands, there is a little information. So if you have only QSARs and some articles, most probably you will not be able to tell for sure. Can endocrine disruptors be classified micropollutants in revision of EU Urban Wastewater Treatment Directive? I have to say I'm quite some time out of the wastewater treatment directives and legislations, but the approach, and now I'm just thinking cloud. The approach of the European Union is very strict towards the endocrine substances. In wastewater treatment plant, you have mostly human endogenous hormones, which are not so stable, but you have there also this ethinyl estradiol, for instance, which is more stable. But this is already somehow managed in the risk assessment of the pharmaceuticals, the concentrations which is in the environment and which is produced. I think it could be an issue as you have this one substance, one assessment. So it could be done, but I don't know. I don't know. What about the proposed REACH revision? Will there be a way to avoid animal testing? I don't know. I don't know. I mean, if you decide to classify as endocrine disruptor, you can probably avoid the testing. And in the I don't see currently the way how to avoid the animal testing, but there is a lot of research done on the new approach methodologies. I think especially the neurodevelopmental study, which I saw very nice research, like in a way of QSARs. This will be very important if this get published and if this gets approved by the authority, that it's enough to say that the substance is not endocrine disruptor. Okay, so any thoughts on how to deal with ED assessment for very complex substances like UVCBs, some natural oils, extract solvents, petroleum products? Yeah. Okay. So in past, I was responsible for some like nature products, extract from the plants, and what's in the CLP guidance is that they want you to evaluate all the substances in the mixture, which is of course impossible when you have UVBC, so unknown variable composition. You cannot. Very often you don't know all the substances. They are variable, and there can be thousands of them. So I think it's impossible to do the evaluation, even the light screening for all the substances. So I think this is very difficult to be handled. And also you can think about a Green Deal. A Green Deal is saying that there should be green solutions like plant extracts and so on, but then they publish guidance which is actually making almost impossible to do the assessment for such substances. So good luck for this. I mean, I saw several approaches how to handle this. You can somehow group two substances, so you do less assessments. So you can have 50 substances very similar, and you do assessment for these substances. But you should evaluate all the substances in the mixture. That's the requirement of CLP. I think it's almost impossible to do so, but it's the requirement.
There is one final question in the Q&A regarding avoiding animal testing, and then there were multiple hottest topics from the registration questions, which will probably sum up.
Yeah, I was replying to this one, but I think I can add that there are a lot of ways in the guidance how to decide that the substance is endocrine disruptor, but it's not so clear that applicant can use the same approaches to say that it's not endocrine disruptor. So I don't know. And I didn't see the REACH review, so I just heard the stories about it and so on.
From the most frequently asked questions, do you think any one of those was left unanswered? I mean, currently the one that I'm showing over.
Okay.
One takes.
So,
I'll give you a
Katrina,
minute.
how to assess biocides for ED properties to non-target organisms? Okay, yeah, that's an interesting question. Like for biocides, plant protection, you will have the assessment according to the plant protection biocide regulation, the assessment will say it's not endocrine disruptor. On the other hand, you need to follow CLP, which is the regulation which is above all the other regulations. So basically, you should evaluate your product according to CLP. So that means that you should basically do the assessment again according to the CLP rules. And in the CLP rules, it's directly written that if the substance is not classified or considered endocrine disruptor, in biocide plant protection, it can still be endocrine disruptor 1 under the CLP. So the CLP guidance is much more broad, much more substances can become endocrine disruptors than in biocides and plant protection. So you need to do the assessment again. I don't know what would be the other way. So with the biocide plant protection, you already you did the literature search, but the search can be kind of targeted. So you need to look again for non-EATs in this search and check for the non-EATs properties in the dataset of biocides or plant protection product. Okay, so that's one question. So then, Sylvia, do you know of any similar attention being focused on these substances outside Europe? Will they be included in GAS? So Europe is really trying to include this in GAS, which is a global harmonized system. On the other hand, I think some countries are not really in favor. So it will depend on the negotiations of European people with the people from outside of Europe. So I don't know how this will develop. Graham improved my understanding of study data interpretation when classifying endocrine disruption. Yeah, so you can look into the ECHA Guidance, and there is a lot of things which are considered indicative what's endocrine disruptor or endocrine active effect. And also we can read the guidance, which is the CLP guidance. There are, for instance, on the thyroid pathway, there is much more way of interpretation added. And also the OECD 150, that's data sets of a lot of guidances and interpretation and talking about endocrine activities. So you can check this.
Okay, thank you, Adam. There are 2 more questions in the Q&A queue.
Okay.
I think these will be the last ones, or do you want to?
Yeah, I think we are 15 minutes after the schedule, so we can do last 2 and.
Okay, let's do them. So the first one I think relates to the one asked in the registration and answered by you a few minutes ago. Katerina agrees that it is difficult to test all the components of an essential oil, but for example, to do valid AMA and FSTRA tests, it's needed to ensure monitoring of the concentration of components of the essential oil in the water through which the UVCB compound is introduced. As it is an oil, its solubility in water is limited. Thus, validation of this method for water analysis is challenging. Do you believe it should be done by introducing the evaluated compound via feed?
Okay, so that's a very yeah. I think I would need more thinking about this, how to, and it would be good to kind of check more information about this substance. because you can also dilute in some compounds to be able to dilute in water the substance. But maybe you can write me an email and I can think about it a bit more. But in general, there are a lot of challenges in the testing. So very often you need to adapt the standard test to be able to do the testing. I think I also enjoyed quite some substances which were not soluble in water, or very specific substances which were tested, like test of gas for in vitro estrogenicity, what I was studying monitoring in past, and so on. So there are challenges. And very often the authority is not so happy about not having the study. So the conduction of study is on the applicant, and the problem that the study is not there is also very often problem of applicant. On the other hand, to be fair, I also met several very nice people at authorities, and they were often able to somehow help to deal with these issues. So maybe you can also try to discuss this with the authority. And I think if it is possible, it's always very good if you are somehow adapting the test from the standard OECD procedure that you discuss with the authority, that they agree with the adaptation, so they later on are able to accept this test when you submit it, because it's a vertebrate test, so it's good to do it good.
I think you've touched upon this, but if maybe you want to reiterate, this will be definitely the final question of the webinar. The question reads: how about the weight of evidence using in vitro quasars and read across in order to avoid animal testing? At least a good read across may still be a viable alternative.
Yep. Yeah, I think it can be a good way for applicants, for industry to actually somehow agree between themselves in consortia, or even across several consortia, because some substances you can group them, and when you group the substances with the similar toxicology, similar effects, then maybe you can use the data already generated for some substance to do read across for other substances. So I think definitely a read across is a very good way how to lower the costs of the studies, and how to do the assessment much more solid. So if you agree with more companies with similar compounds, your competitors, on sharing the data, and that could save the animal testing. It can save the time, and read across definitely it's I think a good approach. But it's about using the animal data, but maybe the animal data which were already produced, or animal data which can be produced for much more substances than for one.
Okay,
So,
with this, I would like to conclude this webinar. Thank you a ton, Adam, for hosting it. Thank you to everyone participating and attending and participating as well. We will definitely be hosting another one with the current ECHA guidance on CLP being very, very freshly released. ECHA has their own webinar in a week on 21st of November. And after that, we would like to also make a more narrow profiled deep dive into the guidance from the regulatory perspective.
okay, so thank you for your attention. See you in some future webinar, or if you have some questions about endocrine disruptors, you can send us email. If you have some substances you want to help with, you can also send us email, call us, and see you some next time. Bye.
Thank you again. See you around.